before yet.
It wasn't set up?
It's set up, but I need to.
You're just going to start.
Okay, but how do I take off? It shows that it's recording. I don't want it to be recording right now.
Can you give us an overview of what you do at Regeneron?
Thanks. I will do that. Before I do, let me just say thank you to Rabbi Zalman Mendelssohn for inviting me into the Chabad of Wyoming. I would love to be there in person and discover what it's like in real life, but this is the next best thing. And thank you very much to Mr. Leon Clark for putting me in touch with Rabbi Mendelssohn. And Leon, it's good to see you. Wyoming accent is developing nicely over there. That's good to hear. And finally, let me reiterate what Rabbi Zalman said, that these words, this learning should be Leilui Nishmata Hanifteret, who passed away sadly today. So with that said, Leon, what do I do at Regeneron was the question. So Regeneron Pharmaceuticals is what's known as a biotechnology company. It's about 25 years old. A biotech company is a place that seeks to use next generation and advanced molecular biology tools to make new therapeutics for severe human diseases. We work across the spectrum of human disease, and as the pressing health issues of the time might change, so we might also change our focus to try and address the most pressing issues of the time. And I'm sure we'll come on to that subject today. Now, it's a company of about 6,000 or 7,000 people. What do I do? So what my group does is we take new therapeutics that have been designed and you could say invented by our scientists. And what we do is we put them into human beings for the first time. So first of all, new therapeutics are developed in the dish, on the lab bench, if you like, and then they're put into animals, tested in animals to see if they work and to see if they're safe. And eventually, at some point, somebody's got to take those molecules and put them into human beings for the very first time to see what happens. And that's what I do.
Regeneron began clinical trials of its rheumatoid arthritis drug, Kevzara, the generic name is sarilumab, on COVID-19 patients. What kind of results have you seen, and what's the next step from that?
So, COVID-19 pneumonia is a type of pneumonia that in some ways is similar to other types of pneumonia. What is pneumonia? Pneumonia is an inflammation of the lungs caused by an infectious agent, in this case, a virus. The lungs get inflamed and people can die because the lungs are the organ in the body where oxygen comes from the air into the bloodstream, and the carbon dioxide that our bodies make diffuses out into the air through the lungs. That's breathing. When the small air sacs in the lungs become inflamed, they tend to fill with fluid. That stops the oxygen and that stops the gases transferring, and that can prove fatal. First, you get respiratory failure and you can die. So what is inflammation? Inflammation is the body's natural response to an attack from a foreign microbe. The purpose of inflammation is to eliminate that microbe, defeat that microbe. I compare it to a war between two armies. Now, the thing about a war is the area in which the battle is being fought tends to get destroyed. So if there's a battle in a particular village, after the battle, the village itself tends to get completely destroyed. And it's not all by the enemy. A lot of the destruction is from our own army. So we need to blow up a building so that we can get our guns on the enemy, for instance. Or we need to dig up a road so that the enemy cannot come down that road. That's exactly what inflammation is. Inflammation, in other words, itself, while you're trying to defeat the virus, inflammation itself can stop or prevent the body's organs working normally. So there is a balance between too much inflammation and the body's response to the virus ending up killing you, and then too little inflammation, and then the virus gets to take over everywhere, and it will end up killing you again. So it's a very fine balance. And early on in the COVID pandemic, there started to be some evidence, at first just describing what the molecular basis of the inflammation was. Now, inflammation is a very basic part of biology. It's very important for life because we're constantly exposed to microbes. And we have a number of different pathways of inflammation. We don't just rely on one molecular pathway to get inflammation, to fight microbes. We can fight them through so many different ways. And so early on, people were analyzing blood samples and fluid samples coming up from the lungs and analyzing them for which molecules that cause inflammation seem to predominate. And the fact is that the word you most heard during those early phases of discovery was the word storm. In other words, this was a storm of inflammation. Every single pathway you can think of, that's what was going on in COVID pneumonia. Now, it's very difficult to fight a storm because if you had drugs that would switch off every single different type of inflammation, you might overdo it. So what we were looking for was evidence that one or two particular pathways would be important. And we particularly got interested with a paper that came out of a Wuhan hospital describing 14 patients who had been treated with an inhibitor of an inflammatory molecule, a molecule that causes inflammation, called interleukin number 6. There are many different interleukins, and this one is interleukin 6. Sometimes people call it IL-6 for short. We were interested in that one because that's one of the molecules that causes rheumatoid arthritis. We already had a drug that blocks interleukin 6. So if it was really true that a drug that blocks interleukin 6 could help COVID pneumonia, that's something that we could bring to the public very quickly. The reason we could bring it quickly is because we had a large stockpile of vials of this stuff. And we were prepared to distribute them in the event that they would help COVID. Now, in the paper from China, it turned out that people who had high fever, people who had low oxygen levels in the blood, and people who required to be in hospital, all 14 of them, after they had had one infusion of an anti-IL-6 inhibitor, their fever went away, and they were all discharged from hospital within a few days. So that was encouraging, but it doesn't prove anything because all of those, one, that's not very many people, just 14, and two, all of them had the same treatment. The thing about a science experiment is to split up the people and to have comparison. So we designed a study to be carried out in the New York hospitals. And that study was to compare a group of people getting our IL-6 inhibitor, sarilumab, to a group of people just getting a saltwater infusion. So no one would know whether they were getting the real drug or what we call the placebo. And we figured out that if we were going to do it properly, we couldn't do it in 14 people. We were going to have to do it in hundreds of people to really get a good look at what was going on. So we did the experiment, and unfortunately, it was very easy to collect hundreds and hundreds of people because there were so many sick people in the New York hospitals at the time. However, we found results that were significantly different from the results in the Chinese publication. So what we found was that there was essentially no difference between the placebo patients and our patients. And I'm just simplifying it. If you look hard at the data, you can find potential differences. But big picture, there was no difference. I'll give you an example. How many people died? So in our experiment, the proportion of people who died was around 30%. And essentially, that was more or less the same whether you were on the drug or you weren't. I'll give you the numbers. If you were on the placebo, the death rate was 27%. If you were on the low dose of the sarilumab, the death rate was 36%. It looks like it's a bit worse, right? It looks like it's even more. If you were on the high dose of sarilumab, the death rate was 23%, a little bit less. Now, all these differences of a few percent, they're all within the margin of error. So essentially, what I think is that these death rates are the same. So there's no dramatic, obvious effect of sarilumab itself on COVID pneumonia. While that was disappointing, it didn't put us off carrying on with our attempt to help with the COVID problem. So we are taking a further look at a larger group of patients treated with sarilumab, and we'll have the final results of that soon. But meantime, we've designed some new therapies to attack the virus directly.
So I've heard a lot about plasma treatment and all this. Can you give us an overview what plasma treatment is?
Yeah, so to understand what plasma treatment is, we have to talk a bit about the immune system. So I said before that inflammation is the body's response to an attacking organism. One of the many parts of inflammation is what we call the adaptive immune system. And one of the things the adaptive immune system does is it makes these proteins called antibodies. They are Y-shaped proteins where both prongs of the Y can bind to, in this case, the virus, okay? And the other prong can recruit other immune cells that can come and kill that virus. That's basically what an antibody is. Antibodies are probably the big reason why once you've had a virus, typically you never get that virus again. Take the measles. Once you've had the measles, the reason that you never get the measles again is not because you never meet anyone again who has measles. It's that if you get infected with the measles, your body can instantly destroy those measles because of the antibodies it still has. So once you've had a virus once, you keep making those antibodies for the rest of your life. Where do they live in your body? In the blood. they live in a compartment of the blood called the plasma. The plasma is nothing to do with television screens. The plasma is simply that part of your blood that is left when you take out all the cells. So it's the non-cell component of blood. That's where the antibodies live. So when people say plasma therapy, what they're really saying is antibody therapy. We will wait for some people to get better from COVID. They will have antibodies against COVID. They will donate blood. We will take the plasma from that blood, and therefore we will be able to treat sick people with those people's plasma. So the sick people will then have those antibodies that can attack the virus.
That sounds kind of simple, just switching blood. Like, what kind of obstacles and complications are there that make it kind of more difficult, or why haven't we seen it everywhere already?
It's a bit like blood donation. You have to get donors to provide the initial whole blood, but then you have to process that whole blood. So you remove the cells, that's what you do first, and then you're left with whole plasma. But whole plasma has a ton of stuff in it other than antibodies. You're really interested in the antibodies. So what you want to do then is you want to purify out those antibodies by passing that plasma over these special columns that pull out the antibodies. Then you end up with and then you get that from each donor and you throw it into a big pool that is known as pooled immunoglobulin. You're right. If it was so simple, we would use it for lots of viruses, right? Well, it's not used very much. Where is it used? So it is used for hepatitis A. Hepatitis A is a nasty liver infection virus that people get when they go usually on vacation to tropical countries. And you can take an injection of hyperimmune hepatitis A that's collected from people who've had hepatitis A. It only lasts in your body for a few weeks. And during those few weeks, you'll be immune from hepatitis A. And that covers you for your holiday or your vacation. Other than that, the only other viral disease where it's given is a viral disease we don't see a lot, but we all know about, and it's pretty scary, called rabies. You catch rabies from the bite of a dog who has rabies. Rabies infects the brain, and it affects the part of the brain that controls anger. So the dog with rabies is an angry dog. If you go near that dog, you may get bitten. You then may get rabies. You then may get angry. You get a brain inflammation and you will die. If very early after the dog bite, you can give people pooled immunoglobulin from individuals who survived rabies themselves, you can ameliorate the cause of rabies. So it works in a couple of instances, especially when someone's taken the trouble to collect the plasma from people who have got better from that disease, enriched the antibodies, pooled it all together, and are going to give it.
What kind of place do you see it in the overall response to COVID? Do you think it will be used a lot, or what advantages and disadvantages does it have with regards to other responses and solutions?
Well, let's assume it works. So first of all, there are trials going on of plasma therapy. So we don't know if it works yet. There are lots of viruses that apparently it doesn't work for. That's why it's not used that much. But let's assume that it does work. So there are various settings you could use it in. Let's take the most serious and go milder and milder and milder. So the most serious setting is somebody who comes into hospital with failing lungs. You could give it as a treatment to that person. Their lungs are full of the virus. They haven't developed any antibodies themselves yet. You give them somebody else's antibodies to attack that virus, help them clear the virus, then the inflammation will go down because there will be no virus left. Okay, now the next less serious case is somebody who's coming into hospital with non-severe COVID. Their lungs are not failing, but a proportion of those people go on to have the severe pneumonia. So we've all heard these scary stories of people feeling absolutely fine for more or less 10 days, and then suddenly on that 10th or 11th day, within a couple of hours, deteriorating and dying. So if you gave somebody that might happen to a treatment like that at day 2 or day 5 or day 8, you could prevent them getting into respiratory failure in the first place. Now, the next group that you could give it to is people who haven't even really got any COVID symptoms at all, but are contacts of people with COVID. So we heard a very sad story in the news this week in London. There was a ticket collector on the trains at Victoria Station who met and encountered an aggressive person who was not feeling well, who was feeling abusive, and who spat at this unfortunate ticket collector, who 4 to 5 days later fell ill with COVID and unfortunately died 9 days, 10 days after that. Now, if that person had had plasma therapy on the day that they were exposed to the COVID, before they ever got any symptoms at all, you could actually prevent them getting the disease at all. And then the final group that you might go for is, say, healthcare workers. They've never had the disease, they're not immune, and they have to go and work in a COVID hospital for a shift of a month, 30 days. You could give them an injection of that stuff at the head of that time, make them immune for that few-week period that they would be working with the COVID patient, and they wouldn't even get infected in the first place.
So on to vaccines. What factors affect the vaccine development for different diseases? Why, for some, do we have vaccines and others we don't?
Yeah, so what is a vaccine? A vaccine is when you safely fool the body into thinking it's infected with that virus so that it develops antibodies to the virus. How do you fool the body into thinking that it's developed a certain virus? Well, the first ever time this was done was in the 19th century. It was noticed by a doctor called Edward Jenner that people who milked cows, and it was a woman's job in those days, so they called them milkmaids. He noticed that milkmaids never died of smallpox. Smallpox was a virus that killed a lot of people in the 19th and even 20th century. And if it didn't kill you, it scarred your skin because it caused these boils all over your skin that were scarring. I remember as a child, you saw people in the street when I was growing up, older people who had large pox all over their faces. You could tell when someone had recovered from smallpox. And I'm old enough to have been in the generation that were vaccinated against smallpox. So this led to the eradication of smallpox. It no longer exists in the world. Now, this all started with Edward Jenner, who noted that milkmaids who milked cows never got smallpox. The reason is because they got a cow virus called vaccinia, from the Latin word for cow, vacca. So The vaccinia virus is similar enough to the smallpox virus that if you've been exposed to vaccinia, you generate antibodies to vaccinia, that will kill smallpox. He then was able to purify the vaccinia virus, which is a cow virus that doesn't really harm humans. And he injected small amounts of the live vaccinia virus into people. And that was the first smallpox vaccine. And it was the first vaccine. In fact, that is why vaccines are called vaccines, because the first one was from cows. So the most obvious way to try and vaccinate someone is to find another virus that is harmless, but looks like the virus you're trying to treat. It turned out that pretty much never happened again. That whole smallpox vaccine thing, it never happened again. It was just the first time. So since then, people have started doing a variety of different things. The first thing they did was what they call attenuate a live virus. So take the virus itself, and they do this with measles. So the measles vaccine, or polio, you could take the virus itself and you could do things to it to weaken it. Like, for instance, you could shine ultraviolet light on it. That would weaken it enough that when you injected it into the person, it wouldn't cause the disease. It would just cause a local bit of inflammation and swelling in the arm where you injected it. And that would be enough to cause the antibodies to be produced. So that was the second step. Then the next step was they purified viral proteins. So now we're getting into the molecular biology era. Purify viral proteins and inject them into the arm. When they started doing that, they found nothing happened. You just put a viral protein in the arm, you never get antibodies. Why? It turned out you need some inflammation to get the system to produce antibodies. And that's when they discovered adjuvants. An adjuvant is a pro-inflammatory signal that doesn't even need to be from that original virus. It could be from somewhere else. A pro-inflammatory signal, just stimulate the immune system enough so that when you put the viral protein in, you get an inflammation and you get an immune response. One of the early ways of producing adjuvants was they got the bug that causes tuberculosis, killed it, crushed it up, and that was the adjuvant. They also noticed early on that alum crystals have an adjuvant property. So alum is an adjuvant. Nowadays, there's many sophisticated forms of adjuvant, and that's why vaccine development has progressively got more and more sophisticated over time. The latest development, which brings us right up to date with the advances in molecular biology, is instead of giving the protein from the virus, you give the DNA or the RNA that makes that protein. So ever since the 1950s, we've known that the proteins that we have in our body, that's the actual substance of our body. It's mostly protein. All of those proteins are made from the genetic code that we have, that's DNA. And there's an intermediate between those two, which is called RNA. So if you give someone some viral DNA or RNA, not enough to reconstitute the whole virus and infect them, but just enough to make a few of the proteins and inject it, the human cells will take that DNA or RNA and make protein out of it. And that could be enough to cause an immune system, an immune reaction. So with COVID, what we're finding is most of those different ways, from the very traditional way right up until the most modern ways, they're all being tried. And so we will see whether we will get a vaccine. Well, every single one of the methods I described is being tried.
We've heard a lot about importance of herd immunity and also the necessity of social distancing And these kind of seem to me to be mutually exclusive. The one suggests we all sneeze in each other's faces, and the other suggests we stay 6 feet apart at all times. What's the right approach?
You're absolutely right, Leon. It is an opposite thing. And I remember that when we were young, when there was far less vaccinations than there are today, thankfully there was vaccinations against the most dangerous diseases. So for instance, diphtheria. All babies today get a diphtheria vaccine. So we don't know what diphtheria is. You know, it's just a word. But my oldest uncle died at the age of 10 years old from diphtheria. And then a few weeks later, his father died of diphtheria. And my grandmother lost her eldest son and her husband in a few weeks of each other from diphtheria. She remarried my grandfather and she had another 2 sons. So you would not want to go for herd immunity when there's a diphtheria epidemic or a cholera epidemic or Ebola virus epidemic. But if you've got something that could kill you, but it's much less likely to, like measles or chickenpox or German measles, then there may be an advantage to spreading that around. Now, take those ones that I gave you the example. If you get those diseases as an adult, they can be much more severe. If you get mumps as an adult, it can make you sterile, for instance. But if you get them as a young child, they tend to be less severe. Maybe because the childhood, the inflammation that a child can produce is less than an adult. So it's better to have those diseases as a child as an adult. So if you did not have, now all of the examples I gave you, there are now vaccines. But in the old days where there was no vaccines, if one of us would catch one of these diseases at school, our mother would actually make us sleep in bed together with that brother or sister so that we would all get it, because it was thought, at least you have it when you're young, it'll do less damage. So if you've got a situation where there is not herd immunity kicks in when about 80 to 90% of people are immune already. The other 10% are protected. If you've got a situation where you're close to that, where it's relatively mild, you know, you can imagine that you might want to be in a situation where you might want to expose yourself. But the coronavirus situation doesn't feel like that at all. First of all, it was unknown how many people it was going to kill. And then when it arrived, we discovered it really could kill you. And especially if you had lung disease or certain other diseases, or you were elderly. So, you know, a lot of us who are elderly or have certain things, we were going to make sure that we were not going to expose ourselves. And we started doing social distancing very, very early on.
This is a last question, but it's a good one. You ready? In a deal criticized in the New York Times, the government has agreed to fund 80% Regeneron's development costs for COVID treatment, without requirement that the treatment be affordable. Additionally, Forbes listed the 2 CEOs of Regeneron in a list of 10 executives who have already gotten very rich off the COVID virus. The public perception of the pharmaceutical industry is already far from good, and this profitability, while the average person is struggling, does little to help that reputation. To what extent do you feel these criticisms have merit, or is the industry misunderstood?
So before I answer the question, let me just tell you what Regeneron's COVID treatment is, because I didn't get to do that. So we have a program called anti-spike. Spike is that part of the virus that sticks up from the surface. That part of the virus binds to the human cell, and then the virus infects the cell. So we have mice that we genetically engineered. They look like mice, you wouldn't know that they're part human. The human part in them is the immune system that makes antibodies. So normal mice have an immune system. They make their antibodies. They make mice antibodies. We made chimeric mice that have a humanness to them, which is their immune system. So when we put the spike protein into those mice, we get a human antibody out of that. The other thing we did quite early on is we got blood from Singapore, from patients who had recovered from COVID, and we analyzed the antibody producing cells, and we found which cells were producing antibodies that seemed to block the spike. And we've made those antibodies too. So we have now got 3 very promising anti-spike antibodies. You don't have to make them. So it's not a virus. We can, and it's not plasma therapy. It's between the two. Plasma therapy, you have to get blood from people. Vaccination, you give to the patient, they make their own antibodies. Here we've engineered an antibody that we can make at massive scale in fermenters. It's the same antibody and everyone can get it in an injection. So that's the latest thing that we're doing. Now, to come to your questions, your last question first. I think I'll do the questions in reverse order. The last question was, has this whole thing made pharma and biotech look worse in the eyes of the public? The answer is that pharma and biotech already are one of the least well perceived sectors of society by the public. People think that the people who work in my industry are worse than a lot of industries, worse than the entertainment industry, worse than bankers, worse than lawyers, worse than accountants. We're generally considered bad. And I don't think that that's got any worse in the latest thing. In fact, I think it's got a bit better. I think my impression is people can see that we're all trying to solve this problem and not just carrying on working on the things we were doing before. You know, I can tell you that in our company, you know, we have shelved a lot of active projects in other diseases that we were working on, but not while moving to COVID, but not completely. So I know a lot of people who are working 12, 15 hours a day doing the COVID stuff and trying to keep their other stuff going on. I think people, I don't see a lot of negative press. I may have this wrong, but that's my impression. Now I'm going to jump to your first question. Now, there is an organization, an arm of the United States government called BARDA, B-A-R-D-A. Has anyone heard of it, and can anyone tell us what that stands for?
No, please go ahead.
The former leader was grilled today by Congress.
Right.
Exactly, Rick Bright. So let me tell you about BARDA. It stands for the Biomedical Advanced Research and Development Authority. Let me just find my Zoom again. There we go. I never remember what it stands for. That's why I had to look it up. What do? They were formed to fight not just pandemics, yes, pandemics, but also bioterrorism. The US government realized many administrations ago that it needed an arm to protect itself from potential bioterrorism and pandemics. It's turned out that bioterrorism, the sophisticated molecular manipulation of infective organisms to harm the population, is beyond the terrorist community. So we really haven't seen, barminan, a lot of bioterrorism. However, we have seen pandemics, and we're seeing one now. And that is also BARDA's job. Now, Regeneron was one of the companies who several years ago realized that we could make antibodies to neutralize viruses like the coronavirus. But we didn't realize it just yesterday when we picked up the New York Times and read about coronavirus. We have been doing this for years. So how many people have heard of the Middle East Respiratory Syndrome or MERS? I see some hands. So that was also a coronavirus. It's actually very similar to this coronavirus. It's obviously not quite as infectious, so it just remained affecting certain population in the Arab Gulf states from time to time. And it's killed a few thousand people, but it has never gone pandemic. It's always just been epidemic. We have an antibody that inhibits MERS very nicely, but there's not a lot of call for it because MERS hasn't been much of a problem. But BARDA noticed that we were working in that area. The next epidemic that happened after that, and I think now we're talking about 5 to 6 years ago, is the Ebola virus pandemic. Who remembers that? Now, I called it a pandemic. That's wrong. It's an epidemic, and it affected the predominantly East Africa, and particularly the Democratic Republic of the Congo. We developed, within a few weeks, human antibodies against the Ebola virus. And BARDA at the time said, that's interesting because nurses going to Africa and coming back may give this to other people in America. In fact, a few nurses did come back and they did get sick. Remember that? Now, thank God they didn't actually pass it on. So it never left the African area that it was in. However, BARDA started thinking, you know what? Maybe we should have a stockpile of anti-Ebola drug. So they called up a few, and the people who were really concerned about Ebola was who? The WHO, the World Health Organization. The World Health Organization found 3 companies that had anti-Ebola treatments. There was one called MAB114, there was one called ZMapp, and there was one called Remdesivir, made by Gilead. Anyone heard of Remdesivir? That's the drug that's just been approved in coronavirus. It wasn't made for coronavirus at all. It was made for the Ebola virus by Gilead. So they started a trial in the Democratic Republic of Congo at the height of the epidemic of Ebola. And no one got a placebo. Ebola's too serious for that, no placebo. But you either got ZMapp, 114, or Remdesivir. We arrived a little bit late on the scene. BARDA introduced us to the WHO. We managed to persuade them to add in Regeneron EB3. There was a cocktail of 3 antibodies, Ebola, we called it EB3. And so there were 4 things in that test. Now I'll give you the results of that test. By day 14 after treatment, so a patient would come to a site with the Ebola virus already sick, right? They would get one of these 4 treatments. And by day 14, incidence of death, Remdesivir, 50%. Now that's much worse than coronavirus. 50% dead in 2 weeks. ZMapp, 50%. Regeneron EB3, 30%. So which is better, Remdesivir or the antibody that neutralizes the virus? In the case of Ebola, it's clearly the antibody. It clearly saves lives and has a much bigger effect. In fact, Remdesivir is considered not to work in Ebola at all. Gilead shelved it. What we did with EB3 is we submitted it to the Federal Drug Authority for approval. Once it's approved, BARDA will stockpile it just in case there's ever an Ebola problem. They will, with the money we get from that, we can provide it to African countries who have no money for free. So at that time, BARDA said, you guys are good at making antibodies. This is years before coronavirus. I'm talking 3 years ago now. They said, you're good at making antibodies. We will enter into an agreement with you where you will make these antibodies for us. We'll make a list of the high priority things. So we made a list of the time. You know, it had things like some bugs you may or may not have heard of, MRSA, you may have heard of that one. Lyme disease, that was on the list. It didn't have MERS. They said, you know, we're not so interested in that one. Not many people getting that one. There was a list we were working on. But critically, in that contract with BARDA, it said, in the event of an emerging epidemic or pandemic, we contract you in advance to work on that one. And in the contract, that was what they were getting. What did we get? Well, they were going to pay for 80% of the development in advance. So I think it's a pretty good arrangement for the American people. BARDA knew that it needed to move quickly in the event of a pandemic. So it pre-arranged with companies who had a proven track record of working in this area. Now, and that's what we did. Within a few days of the news coming out of China, we were already making our antibodies. Look, I think it's a good thing. And I also think it's a good thing that a whole bunch of other companies have jumped on the bandwagon as well. You know, great. You know, if everyone gets on it, there are many shots at goal. We win Shalom al Israel, as we say. But the arrangement we had with BARDA, as far as I'm concerned, was something that the American government did that was very sensible. And we were disappointed when BARDA director was forced out of his position. But he testified in front of Congress today saying that his firing was a political move and not a scientific move. In the meantime, BARDA is being run by his second in command, who is also a very good guy that we have a very good relationship with. So the projects are still going ahead just as they did before.
Dr. Harari, thank you so much for that wonderful presentation. And please put your hands together for the wonderful questions from Leon. That was amazing. Thank you so much. This was so clear, so simple to appreciate, so simply defined, and yet so wonderfully complex and detailed and wholesome. It was fantastic. Thank you, Dr. Harari. We have some questions from our audience that I'd like to be able to get to. Typically, we have people write them in and then I will ask the questions myself, but we haven't had that many questions. We've had maybe, I don't know, less than 10 questions. So what I figure is maybe we'll get some of the people who have asked the questions, they've typed them in, that they can unmute themselves and ask the questions themselves. So I'm going to start off with Cheryl Hutt, who's a very dear friend of ours. She asked the question was Sirilimab, something related to patients, cytokine storm. If you can ask your question related to the question that you typed in for Dr. Harari live, and just state the question. If you have a second to just describe your medical background so Dr. Harari can appreciate that, that would be great.
Okay, well, I'm a dermatologist, have been for 40 some odd years, and worked for many years at Columbia. Now I'm an emeritus there, and continue working part-time at the Bronx VA. So, and my brother's a pulmonologist. So I was interested in the Sirilimab studies, and interested because number one, you cited a very high mortality rate. So I'm assuming that this was given in only very critically ill patients. And might it not be, if we could tease out some of the data at some point to see how patients reacted to these drugs, yours or any others, given at various times, might it not be more effective given earlier rather than later? And that was sort of one thing. And the other one was, again, in terms of patient selection, the interleukin-6 drugs should work presumably for people who are in cytokine storm and not who are sick, who are deathly ill from other manifestations of the coronavirus. And so were those patients selected for primarily during these studies, or was it just given to everybody who was sick?
So first of all, Cheryl, I just want to say that at Regeneron, we love Columbia. Our company was founded by two friends who are still 25 years later the chief executive and chief scientific officer, Len Schleifer and George Yancopoulos.
Yes,
and
no George. We know
they
George.
both came out of labs at Columbia. It's great to hear that you know George.
And do you know Christos?
Yes. So so for everyone who
Christos is a
doesn't
very
know,
good friend of ours.
George is the CSO of our company and is a bona fide genius. He's a member of the National Academy of Sciences in the US, which is a very small and elite group of people. And I don't think there's a lot of people in the biotech industry in that group. It's really predominantly people who work in the top universities. And so he's a bona fide genius. Christos is a relatively young person that we have working in our company, and he is the head of our infectious diseases discovery group, and he is the person who makes these viruses. And a nice young man you could not hope to meet. Lovely, lovely guy, and I was just speaking to him today. So coming to your question, so I did give you the mortality rates in the critical illness group, the people who are critically ill. But we studied three groups. Severely ill, that meant you're on oxygen, and you're in hospital, but you're not on a ventilator, you're not on special assisted ventilation. That's the severe group. The critical group, you're on a ventilator or similar. And then there was the multi-system organ dysfunction group. That's the group that's beyond just respiratory failure and now has renal failure or liver failure or heart failure, multi-organ failure.
Where many of these drugs are probably not going to work anymore anyway.
Yeah, we thought the drug might not work, but we can't, humanistically, we can't say, oh no, you can't have our drug, you're too far gone.
No, I didn't mean that, but I mean they may be beyond the time when the drug is going to be effective.
Exactly. So we didn't include them in the analysis. So I have no information on them, although unfortunately, a quarter of the patients we recruited were in that multi-organ dysfunction group.
Mm-hmm.
Of the rest of them, it was 2/3 critical and 1/3 severe, the milder group. It turned out we saw some trends to benefit in the critical group, but not in the severe group. So going forward, we're only going to study the critical patients. Now, we were hoping it might be that we might find a molecular signature, as you say, but it turned out all of these patients, and in fact, everyone we screened with COVID pneumonia had an elevated IL-6 level, which is the molecular protein we're trying to block. Everyone with COVID pneumonia has a high IL-6. So if there is marker of who's going to respond, it's not the obvious one. Mm-hmm. But finally, just to say that if you merge the severe illness group and the critical illness group, overall death rate in that whole group was 10%. And the discharge rate at 28 days was 80%. Now, that's bad. That's a severe illness. You're not going to want to have COVID parties, herd immunity. But it was it's less bad than the original news. When we first, I remember the day we first got the news out of China, the early data then said that you have a 25%, if you're severe, you have a 50% chance of becoming critical. And if you're critical, you have a 75% chance of dying. And
Right.
these numbers are not that bad. So the death rates in real life in New York City are not as bad as, say, Ebola virus or some of the really, really highly fatal
Right.
viruses.
It's not the numbers that Cuomo is quoting of, which are correct, by the way, but of the patients on ventilators. So once you're on a ventilator, your chances of leaving the hospital are about 20%. So, and that may still be true. I've also noticed that the numbers in New York seem to be, in terms of the overall death rates, and which I'm not sure why, seem to be a little higher than elsewhere. New York State overall in the country seems to be up about 5%, where the rest of the country, the individual states look to be about maybe 2 or 3%. I'm not sure why that is.
Let's move on to the next question.
Sorry, yeah, go ahead.
No, Cheryl, you're fantastic, but let's.
I could talk forever.
Dr. Harari, do you want to comment on what Cheryl just said, and then we'll
No,
move
that's
on?
okay. That's not his.
Look, I mean, I don't know about, it's really hard to say whether differences in healthcare cause differences in mortality. What we do know is older people, people who are non-white, people who have certain conditions like high blood pressure. So you could imagine that the proportion of that group in New York City compared to Seattle, for instance, might be in favor of seeing a higher mortality in New York City.
Okay, thank you very much for that discussion. Let's move along to Johnny. Johnny, are you on the call? If you can come live and ask your question, unmute yourself. You asked something, do you believe that the virus originated? Can you come online and ask your question? Unmute yourself, Johnny.
Thank you very much. Thank you. Yeah, I'm interested in your opinion, Doctor, having looked at the virus up close, as to what you think the origin of the virus is, in terms of, was it transferred somehow accidentally by evolving from animals and transferring to humans, or perhaps through from a laboratory somewhere where people were somehow experimenting, or through that way, so not unnaturally, in other words.
Hi, Jonathan. So we've done a lot of genetic sequencing of the virus as it spread around the world. And the basic genetic sequence is the same, that it's a hybrid of a human coronavirus and with a large section of coronavirus from the horseshoe bat. The horseshoe bat there's a large population of horseshoe bats in mainland China, and it's been known for years that they act as a huge reservoir for coronaviruses. Horseshoe bats essentially are very, very infected with coronaviruses, and the splicing section of the bat virus that's coming to the human virus, it's not it doesn't look engineered. It looks like a random event. It's not like when people engineer things, they take sections of a genome, cut those out and put them into another genome in a way that makes sense. Oh, I'll take this section of proteins from the bat and I'll put them in. Like when we made a humanized mouse, you can tell it's not natural because it's just the immune genes that are human. It's neat and tidy. This bit of horseshoe bat that's stuck into the coronavirus genome is completely random. You wouldn't even be able to look at it and say, oh, this is going to be particularly infective now. So I think, and we have seen these events happen before. So SARS, I think it's a mistake to call this thing COVID. This thing should be called SARS-3. And Middle East respiratory virus should be called SARS-2. And then what they call SARS should be called SARS-1. They're all coronaviruses, and they've all come out of random mutation events when different species are cohabiting closely in a way that is unnatural.
Thank you very much.
That was fantastic. We will move along to our next question from Karen Fine. Karen, could you please unmute yourself so you can ask your question? Is there work being done? And if you can ask your question just directly, succinctly, and shortly. So
Sure.
we can as many.
I also wanted to comment, though, on the high percentage in New York versus some other states. I live in Florida, and the Miami Herald and Palm Beach Post are actually suing the state of Florida to get actual numbers. They feel they're being doctored and not fully reported. So some of your difference could come from that. And then my question was, is there a way when we give the antibody therapies, can we prolong the effectiveness and the efficacy so that a nurse is working in a hospital for 2 months, because I am an RN, so I think that way, so that we can prolong the efficacy so they don't have to get another infusion of the antibodies. Thank you.
So, Karen, I said that an antibody is a Y shape, and I talked about the stem. The stem is what controls the duration of it in the body. We've been engineering and tinkering those stems for years to come up with an antibody that has a very long, what we call half-life. The other determinant of the half-life of an antibody is how much it's binding to something. So if somebody's full of COVID and has a huge viral load, the dose of the antibody isn't going to last very long because it's all going to bind up, right? But if you've got a nurse who has no COVID and who's about to go on her shift, then that is going to last a lot longer. So in that setting, we think that one injection could easily last 4 weeks and may last as long as 12 weeks. We're going to start human testing, please God, next month in June.
Thank you very much. We will move along to Professor Saul Silverstein, our very dear friend. Saul, if you can ask your question very succinctly and directly, but beforehand, Saul and Cheryl are husband and wife. Saul served as the chair of the Department of Immunology and Microbiology at...
From which George graduated.
So
I was George's chair. I was also Christos's thesis advisor. But let's go from there. My real question is, there has been no evidence that DNA or RNA vaccines are efficacious yet. How do you feel about that?
I think that the whole vaccine story is impossible to predict. It could very well be. It's proved a little bit random which infections, which viruses are easy to develop vaccines for and which aren't. Yes, we have more modern techniques available now that enable you to rationally design the vaccine and therefore get into human testing more quickly. And that's what we've seen. So the first ones that are coming through are the newer ones, the RNA vaccines. But like you, I have no way of predicting whether they're going to work or not. And I think everyone should prepare themselves for the possibility that there may not be a vaccine.
thank you very much. Saul, do you want to follow up on that?
No, I'm good. I'm in total agreement with that. I'm perplexed by why there's such a push to move into this field when there's been, the DNA vaccine studies have been going on for 15 years now with no success.
Dr. Harari, any comments?
I agree.
Okay, moving along, we got a question from someone who can name himself if he chooses to, but he wrote a question that might be a little bit personal, so I won't name him. He asked, I contracted COVID-19, fell ill on March 21st, and had flu symptoms for 1 week, thanks to hydroxychloroquine and azithromycin. And yet several weeks later and 7 swab and blood tests later, I still carry the COVID and am negative for IgG. I've been in isolation and my wife is fine and negative for virus. Am I still contagious, and how do I get out of the cell? I would just like to comment on that. If you'd like to make yourself known and speak directly to the doctor, please unmute yourself. Feel free to do so. I contracted the virus too, Dr. Harari, and I just recently, as of Friday, got myself tested for antibodies. And while I tested positive for the virus, not me myself, my wife tested positive, my parents tested positive. I was with them in the same home for 2 weeks without going anywhere, 3 weeks, 4 weeks, 6 weeks without going anywhere. And then I went to the doctor this Friday to get myself tested for antibodies because I wanted to donate plasma. And my antibodies came back as negative. I was like, what in the world? You know, I'm in the same house. I had very few symptoms, almost none. That's why I didn't get myself tested. I figured if they have the virus, I have it. If they don't have it, I have no symptoms, I don't have it. But that's essentially surprised me. I wanted to be able to donate plasma. I wasn't able to do so. I won't be able to do so now. Any comments on that, and comments on the question from this audience member who's wondering about his own personal circumstance with the virus?
Yeah, and there's strong similarities in both, because we're talking about the immunity. We could talk about how much of the fact that he only had symptoms for a week really was due to hydroxychloroquine and azithromycin, but that is a tangential issue. I'm going to leave that for now. The question is, why, with these confirmed cases, do we have negative tests? So before I talk about why that might really be happening, just to caveat that all of these tests, both the tests for the virus and the test for the immunity, are being developed on the fly. And some of the companies are doing a better and more reliable job than others. In general, you can get false positives and false negatives with any test, right? We all know what that means, false positives and false negatives. But in general, PCR tests that measure viruses by PCR, which is what all these swab tests are, they are more likely to produce false positives than false negatives. And the same with, from what I've seen, with the antibody tests, they're more likely to produce false positives than false negatives. So if you come negative, so either if you've had a test repeatedly and it always gives the same result, then you can believe it, I think. And also, if you're negative rather than positive, maybe you can believe that too. I don't know. There's so much unknown. My cousin, Mickey, Alav Shalom, he was one of the first people who died of this pandemic because he, along with most of my family, live in the Italian city of Milan, which, as you know, was hit very early and very hard. And he had negative tests, several negative tests the whole way through while he was deteriorating, and he never had a positive test. We clearly had the virus. So one bit of this is the difficulty of understanding whether to believe tests and which tests to believe. But let's just assume that we can believe the tests in this case. Weeks and weeks and weeks carrying the virus and not converting into an immune positive status. Well, there's something consistent there, isn't there? Which is, if you developed antibodies, then you could cure the virus. The fact that you haven't developed antibodies means that you're still carrying on with the virus, right? It hasn't completely cleared. That's kind of logical. You clearly have enough immunity from all the different forms of immunity that you have so that it's not affecting you, you're not ill with it. And that puts you in this difficult situation of being a possibly infective carrier and therefore locked up in your house for who knows how long. And we've seen this before going through history. You know, my wife and I were just reminding ourselves of the famous story of Typhoid Mary. Typhoid fever is a disease that people still get today. It was a disease that killed a lot of people. It could kill you quite quickly, or it could kill you slowly, or you could completely survive it and be immune, or you had people like Typhoid Mary, asymptomatic, no symptoms, but carrying it and infecting others with it. So until this is understood, yeah, you're going to have to be in isolation, I suppose. Now, what are the reasons why you might not have made an antibody? One is that you might, at this stage in your life, depending on your medical history and whatever medication you take, your immune system may be a little bit sluggish nowadays. It could be that. On the other hand, it might be nothing to do with that. It might be that this virus may be quite good at evading the immune system. One of the most famous viruses that evades the immune system is the HIV virus that causes AIDS. You know, that's a chronic disease that no one's immune system is able to clear. Hepatitis B, hepatitis C, good examples of viruses that can lurk around in the body because they've evolved to be very good at evading the immune system. Is coronavirus 19 one of those viruses? I don't think it clearly isn't as bad as HIV, because we do see a lot of people getting better and becoming immune. If it was not the case, then we would have exhausted our supply of healthcare workers already, wouldn't we? So the fact that we still more or less have the same group of healthcare workers that we had 8 or 12 weeks ago tells you that it must be possible to become immune to this thing and be protected from further attacks. But there will be a spectrum of responses, and it's too early to tell whether remaining negative for the antibodies and carrying the virus for a long term is going to be a real thing. So all I can tell you is you're creating new science every day, and every time you go for another test, you are creating a data set of new science that was unknown the day before and is just becoming known today.
Thank you very much for that clear and simple and easy to understand response. Final question that we have written in, and then Dr. Harari, can we maybe have still time for another 1 or 2 questions from the audience if they'd like to just chime in more casually? Would that work for you?
Sure.
So Scott Aaron asks, is it normal to get an ethical approval for drug versus placebo trial, or have ethics committees been more lenient due to the pandemic? If you can comment on that, that would be great.
So that's a very interesting topic, Scott. Normally, you cannot experiment on humans without getting the approval of ethics committees. And that has not changed even during this pandemic. Every single experimental test that's being done on a person has been approved by an ethics committee. Now, sometimes it's more ethical to do a placebo-controlled test, and sometimes it's more ethical to do an open test where everybody gets the drug. And situations like this are kind of in the middle. On the one hand, there's no great evidence that any of these drugs work. So there's no big problem with giving someone a placebo along with one of these drugs, because no one knows for sure that they work. On the other hand, some of these people are getting really sick. So the idea that they're getting a placebo, which we know won't help them, when they could get an investigational drug that might help them, that's kind of scary. What we've seen is that people who are submitting open label studies without placebo, they're getting approval. And we who are preferring placebo-controlled trials, we are also getting approval. Now, something related to your question is not the ethics committees, but the regulators, the Federal Drug the Food and Drug Administration of the United States, for instance, emergency approved remdesivir for use. And the European Medicines Agency is currently moving to do the same thing, emergency approval, based on data that I cannot tell you about because it has not been released to the scientific community to review. So I suspect that when that data is released to the public to review, it might not be of the quality that we are used to, because it is difficult to do a large clinical trial of good scientific quality when you're in a rush. And so I think that what we're seeing is slightly less good quality clinical trials going on, unfortunately. And I think it's good to have clear heads, because just going back to the hydroxychloroquine story, at one point, every single person in the world was going to end up on hydroxychloroquine. It really got a lot of momentum based on open label, non-placebo controlled studies. And eventually people did some placebo controlled trials, and it appears from the latest of those that it has no effect whatsoever, beneficial effect whatsoever. So I think it's important to not panic, keep our heads clear, and still do proper placebo-controlled trials, even though that's difficult in the current environment.
And thank you so much for that, and thank you for the fantastic question. Linda, please unmute yourself. I see you have a question. Please ask your question live. We're moving on to live questions, no more chat questions. If you have a question, unmute yourself, make yourself comfortable, and go ahead and ask. Linda, go ahead.
Is it possible to develop antibodies and then the antibodies become undetectable? And if the antibodies become undetectable, if you were re-exposed, would you be protected by those antibodies? Would they rise to a level that might be protective?
Linda, that is theoretically possible, but unlikely, because antibodies, long after an infection has been cleared, the type of cell that makes the antibody is called the memory B cell. It's an immune cell, but it's not called a memory cell because it lives in our brain. It's a B cell, it lives in the body, but we call it memory because it has immunological memory. It's your body's memory of having had that virus. So theoretically, if the number of those B cells, memory B cells that you had, was low enough, and their rate of antibody production was low enough, you would become undetectable antibody-wise. But when you were infected with the virus again, some of that virus would be presented to those memory B cells, and they would ramp themselves up and knock that virus out. However, there really is because those memory B cells do tend to make low levels of antibody, there is definitely a correlation between the level of antibodies and the amount of protective memory you have. So we all have to have tetanus boosters from time to time. Every 10 years, they say, you're supposed to get a tetanus booster, because this is one of those vaccines that seems to very slowly decline. And once those become undetectable, even if you've had it every 10 years for the last 40 or 50 decades, you still need that booster. And antibodies are a good guide for that. So if there was a vaccine and it was like that you developed antibodies but then cleared them, I think people would want to revaccinate themselves once the antibodies disappeared.
Thank you. That was fantastic. That was fantastic. Any other questions that we have from the audience before we call it a day for those of us who are in Jackson or New York or otherwise in America? and or a night for those of you who are in Europe. If you didn't yet ask a question, we'd like to take your questions first. If you already asked a question, then if nobody else has a question, please ask a second question. Go ahead. I see Yasmina raised her hand. Yasmina, please. Please ask your question. We'd love to hear from you. Open your mic, unmute yourself. Are you able to unmute yourself, Yasmina?
I don't think so.
We hear you loud and clear. Go for it.
Can you hear? Okay, thank you. Well, I'm in Jackson, actually. And I wanted to ask Dr. Harari about your perspective. Could you share with us your perspective on the tribe immunity? How does it work? I mean, there might be venues, maybe more than a vaccine until they're ready to be able to tackle the problem. So, you know, we as community, the collective, can go back to, I wouldn't say normality, but our lives. I mean, you know, the world cannot just stand by until there is solutions with vaccinations. But I wanted to have your perspective, and maybe you can share it with us.
So the question, Yasmina, is how do we get back to normal life? Is that right?
That is the question.
I think what's going to happen here is that we're going to see a sawtooth pattern with the number of cases. It's going to be cycles of lockdown and release. We're just now coming out of the first period of lockdown, and we're beginning to come to deconfinement in the first period. There will be a period of relative freedom over the next few weeks, possibly months, possibly only weeks. And then the number of cases will start to go up again, and the lockdown rules will be restored. Now, hopefully, and that will lead to a second wave. And then there will be a third wave and a fourth wave, as long as it takes to develop, for instance, herd immunity. Now, right now, the latest indications are that in densely built up areas that have been epicenters of the pandemic, such as New York City, only 15% of the population currently are immune. That goes up to 25% in the healthcare workers, meaning even healthcare workers in New York City, 75% of them haven't had the virus yet. And if you take a random sample of people across a whole country, then you are looking at 3% or lower rates of exposure and immunity. So we've got a long way to go before we get to herd immunity. So it's going to be a first wave and a second wave. My prediction is that the second wave is going to kick in towards the end of the summer. What I don't know, this is just my guess, but what I can't even guess is how big the second wave will be. So in the Spanish flu pandemic of 1918 and 1919, the second wave in early 1919 was 5 or 10-fold bigger than the first wave in the summer of 1918. It's really in 1919 that hundreds and millions of people died. So pray to God that the second wave will not be hugely bigger than the first wave. Maybe it'll only be as big as this first wave, or maybe it will be smaller. In the Zika virus epidemic of a couple of years ago, the first wave was the biggest wave, the second wave was small, and that was it. There weren't any more waves after that. So that's how unpredictable it is. And all we can say is, it's in the hands of the Borei Olam. It's in the hands of God, who is the only being who can tell us what will happen, who is determining what will happen, and he's open for us to ask him for a good outcome. And this is what we can pray for.
Thank you, Dr. Harari, for those wonderful words and for that inspiration. Always welcomed here at Chabad. If anyone else has any thoughts or questions that they'd like to ask, please let me know. Otherwise, we're going to move along to some who have asked for a second question. If you haven't yet asked the question but would like to be able to do so now, we have time for another 2 to 3 questions at the most. Glyn, please unmute yourself, or I'll do that for you. You are now unmuted. Glyn Clark, please go ahead and ask your question.
Hi, Dr. Harari. Nice to see you again. How are you?
Mr. Clark.
Very nice to see you. So I guess my question, as you've been up and personal with the virus, is what precautions do you personally take when you're out in the street, going into shops, etc.? What do you think are the appropriate precautions that people should take? Because obviously there's a lot in the media about what people should do. It'd be good to hear it from you.
Yeah, Glenn. So I guess I'm slightly atypical on this. I'm not so concerned about gloves and masks. I'm really into distance, and I'm into hand sanitizer and washing. So I'll go around just like this, but I'll keep my distance. And when I have to touch something or someone that's recently touched, I don't panic. I just know that my hands are now kind of contaminated. And then I get a squeeze of the sanitizer if I'm out of and about, and that's done. Or, you know, if I'm somewhere where there's hand washing facilities, just simple hand washing with soap as well. So for me, it's the distancing and the keeping the hands clean that is much more meaningful than the barriers. Or I don't know what you guys think.
Thank you. That was fascinating. Here I am walking around with my glove and masks. I'll continue to do that because I'm a rabbi, and people wonder, why is the rabbi walking around as if he thinks this is, you know, South Florida or something? Get your act together, put on a mask. So I make sure to do that at all times that I'm in public. Any other questions that we have, first-time questions, please go ahead. We'd love your questions before we call it a day. Final questions. Scott, please go ahead and ask one more question.
Thank you very much, Rabbi. Thank you, Oliver, for your speech. I think you've sort of partly answered it. I was wanting really to ask that as a researcher of drugs, you're perhaps the wrong person to be giving the questions. It seems that the people we want to speak to at the moment are the epidemiologists. But you've answered your views on what the epidemiology may be going forward.
That's right.
No longer a question, more a comment at this stage. So
yeah,
sorry.
Right. It's just a sense that I have rather than, as you say, it's not my expertise. But Rabbi, with regarding to the masks, so have you not heard? I mean, I've read many times that the beard obviates the mask. That if you really want to wear a mask, you've got to be, no, not even a short one. You know, absolutely clean, fresh Gillette that morning. So that's what I've heard, at least, and that's one of the reasons I'm not wearing one.
Well, thank you for pointing that out. I will note that my colleague and friend, the doctor who was at the head of an ICU at a Tel Aviv hospital, who's a Chabad person who has had a beard his entire life, at the beginning of this virus, he spoke to some other rabbis, and as a result of needing to be able to care for the well-being of fellow people in Israel, he shaved off his beard with tears in his eyes, but with a full heart, knowing that he has to do what he has to do in order to be able to save other people's lives. So when it comes to the safety and security of others, we know that first and foremost, even if there's a safek pikuach nefesh, even if there's the smallest doubt as relates to saving and caring for other people's lives, that always comes first. And so as a rabbi, I guess I stay away. I keep my mask on, keep my gloves on, and hope for the best. I do a lot of praying, not much I can tell you. In the meantime, this was really fantastic, Dr. Harari. Your clear, concise way of sharing some of these complex immunological, biological, virological issues was very simple to understand and deeply appreciated. Leon, your questions were fantastic. And with this, we will call it an evening or a day. Thank you so, so much to everyone who joined us for this. Please join us on our upcoming Zoom meetings. Dr. Harari, with a deep debt of gratitude, we appreciate every moment that you shared with us. Our community is indebted forever for your wisdom and guidance. Thank you.
Thank you very much, Rabbi.
Have a wonderful day. Please, Dr. Harari, before we go, final thing. If anyone would like to be in touch with you for whatever reason, how can they reach you?
You can give them my email address, Rabbi.
Fantastic. So email me if you'd like to reach Dr. Harari. My email address is simple to remember, rabbi@jewishwyoming.com. That's rabbi@jewishwyoming.com. Thank you all so very much. Have a wonderful day. Have a wonderful evening. This was fantastic. Thank you.